All right, I think most people, if not everyone's back. So let's get started. All right, good morning. So my name, again, is Ryan Lee. I'm a research software engineer and at Lawrence Berkeley National Lab and the technical lead of the NeuroData Without Borders, or NWB project. I'm very excited to be here, tell you all about NWB and the DANDI Archive and how they drive collaboration and data reuse in neurophysiology. Before I start, I want to take this opportunity to survey all of you really quickly about NWB and DANDI. How many of you, just raise your hand, have used NWB data in some way before coming, or before today? I can see 1, 2, 3, 4, 5, 6, 7. All right, good. That's about a third of you. And also, I want to ask how many of you are going to be working in Python during this workshop? Okay. And then how many in MATLAB? Okay. Great. That's good to hear. Over the years, we've been seeing a greater and greater shift toward Python, and most of our tooling is in Python, so that's just good to know to inform our development. All right. Oh, and last question is, how many of you have generated data and published it on DANDI? Okay. Yeah. We hope to foster a culture of data sharing, and I'll talk to you all about that as part of this. Great. So all of what I'm showing you today is the result of a large-scale collaborative effort across multiple interdisciplinary teams and hundreds of community members over the past 10 years. The instructors throughout this workshop, there are only a few representatives of this large collaborative effort, and we're really excited to have you all be a part of this effort by being here in this workshop. So in this next 45 minutes, I'll first discuss the need for data standardization and sharing in neuroscience and tell you how NWB and DANDI address this need. And then I'll give you a quick look at how to convert data to NWB and share it on the DANDI Archive. Next, I'll introduce how to explore and use data on DANDI, and I'll end by showcasing a variety of community tools for data analysis, visualization, and management that work with NWB data. Now, this will be a lot of information at once, but the goal of this first talk is to give you a broad overview of NWB and DANDI. And later talks over the next few days will go deeper into detail on these topics. So, to start, why should neuroscientists standardize and share data? As I'm sure you know, neurophysiology data are expensive to collect. They're expensive in the cost of the equipment used. They require significant time and labor of grad students and postdocs, and we use animal lives. And so it's our responsibility to maximize the value of this data that we collect. These data are often rich and can be used to answer different scientific questions or develop new methods. And because of this, reusing existing data within your lab with collaborators and across the community can really accelerate scientific discovery. However, making a data set understandable and reusable by others has traditionally been tedious. Most scientists have different ways of organizing and storing their data. It might be a mixture of custom MATLAB and NumPy files, spreadsheets, handwritten notes in your lab notebook. And so collaborating with someone often involves explaining all of your data structures and where to find them in a long chain of emails and meetings. And this is tedious. It's error-prone, and across the field, it's highly inefficient to using this data. Yeah, and ultimately, this tediousness disincentivizes us from collaborating with each other and reusing all this valuable data. And lastly, using other people's scripts and analysis tools, they often require converting data from one custom format to another. And that can be impossible if key metadata is missing, and it can take a lot of time and result in errors. And so you might not even bother trying out new methods or comparing them against each other. And ultimately, this hurts the field and the ability for you as an individual to advance your research. As Ben mentioned, without data sharing infrastructure, neuroscientists often collect new data and build new tools when they don't necessarily need to. And this is bad for efficiency and quality and reproducibility of our research. And ultimately, we want to change the mindset of researchers who are going out starting to try to answer a question from asking what experiment should I run to answer that question to what query can I run on open data to start exploring and answering that question. Many different types of organizations, they recognize the value of data sharing and are establishing rules and expectations around it. This includes government funders such as the NIH in the US. They say now, as of 2023, that all scientific data should be made accessible as soon as possible and no later than the time of a publication or the end of the grant, whichever comes first. Foundations such as the Aligning Science Across Parkinson's Foundation of the Michael J. Fox, the ASAP program of the Michael J. Fox Foundation, require that data, code, and protocols are shared at the time of publication. And increasingly, we're seeing journals, such as the Nature family of journals, require data sharing upon publication. But we all know that sharing and reusing data are still far from the norm. This is a reaction graph, think back to chemistry class, with two states, no data sharing and reuse on the left, and data sharing and reuse on the right. And there's a cost or an energy required to be in the left state. Doing new science can be harder and less efficient without sharing and reusing existing data that might be relevant to your project. And transitioning to the right state, where you can easily share your data and can reuse other people's data, it has a cost. There's this energy barrier, and it takes time and energy to figure out how to share your data and do it well. The funding and publishing requirements I mentioned make it harder to do science without data sharing in the left state, and the prospect of new science and collaborations can make data sharing more appealing and beneficial, advantageous for your work in the right state. But these sticks and carrots, they don't address the energy barrier, the big hump to get from the left to the right. And many of us have experienced this barrier when trying to either use existing shared data or to share the data that you collect yourself. It can be hard to learn a new formats, convert existing data analysis pipelines, et cetera. You might have to figure out what data and metadata about your experiment do you store, what do you name your variables, how do you organize it all, where and how do you share the data, and for the PIs, how do you pay for that data sharing? And for all of you interested in data reuse, how do you find and read and understand all of this data that's been shared by others? Our solution to lower this energy barrier for neurophysiology data, at least, is NWB and DANDI. NWB provides a data standard, which is simply a set of rules for how to organize experimental data and metadata so that anyone familiar with the standard can fully understand the data. NWB also enables a software ecosystem where a large collection of processing and analysis tools can use the same data format for inputs and outputs. And so you, as users, do not need to convert data between various custom formats, making it easy for you to work with this data. Now, the DANDI Archive provides a free, centralized, cloud-based data repository where you can publish and share your neurophysiology data, find interesting data sets shared by others. And DANDI also provides tools to make it easy for you to do all of this. And as Ben mentioned, we often think of NWB as analogous to the MP4 format. It's a common input and output format for videos. And DANDI is like YouTube, a place where you can upload and share your videos for free, search for other videos, and stream it for free. The primary mission of both of these projects is to lower the energy barrier of data sharing as much as possible through tooling, outreach, and dedicated support and events like this. We want to make it as easy as possible for you to standardize and share your data, as well as reuse existing data that's been shared to advance science. Okay, so what do we mean by neurophysiology? Now, we define it as the study of neural activity at cellular and circuit resolution. And this includes electrophysiology, such as neuropixels and patch clamp recordings. It includes optical physiology, such as calcium imaging, voltage imaging, and fiber photometry. And it includes behavior that's often recorded alongside these neurodata, such as video and audio. And in each of these cases, you have the raw data. You have your voltage traces, your calcium images. You also have the results of critical processing steps, spike sorting, image segmentation, pose estimation, that transform that raw data into a form that's ready for analysis. In NWB, you can store both the raw and the processed data, and we encourage you to store both so that you and others can fully reproduce the results of your processing and analysis, can build methods that transform your data, and fully reuse your work. Now, raw and processed data alone isn't enough to be understandable and reusable. The goal of NWB is to store all of this data together with all of the metadata that's necessary to understand and analyze it. So NWB defines a unified data format for the most common types of neurophysiology data and metadata, including all of the neural activity, neural data that I just mentioned, the behavior that's associated with it, but also critically the metadata about the data acquisition, such as the device settings, the targeted brain area, et cetera, metadata about the experimental design, such as the stimuli and the trial structure, and metadata about the subjects themselves, such as its species, genotype, and age. NWB also supports extensions to allow users to add new data types and modalities not covered by the core standard. And all of this data and metadata related to a single experimental session is then packaged into a single NWB file. So one experiment will often have tens to hundreds of NWB files. NWB uses a formal specification language to define over 40, maybe 50 now, structured, specialized neurodata types for all of these types of data and metadata. And each neurodata type has rules on how to name its various elements, what metadata should be provided, what units of measurement should be used, and so on. And this helps to ensure that an NWB file contains all of the data and metadata necessary to understanding and analyzing this data in a standard, accessible way. Under the hood of an NWB file, you'll find a hierarchical organization of data and metadata stored in specific places that are both human readable and machine readable. So here's an example, a very simple example of an NWB file with extracellular electrophysiology data. In the top level general group here, you can see information about the experimental subject, the experimenters, and the locations of the electrodes. In the acquisition group, you can find raw data. So here we have raw voltage data that's stored in an electrical series data type. And this includes a 2D array that's timed by electrodes. It includes the timestamps for each electrode, the electrode's axis, which links to the table of electrode metadata I just showed in the general group. And the data type also includes the metadata necessary to interpret the raw array, such as the units of measurement, the sampling rates, and any conversion factors used. You'll find process data in the processing groups or the units table. The units table contains information about any spike sorted units, including the spike times, the mean waveform, and other features of each unit. And then sometimes it also includes which electrodes each unit was observed in, which again links back to that electrodes table. So yeah, again, all of the data and metadata for a single session are stored in a single NWB file. And that NWB file can be written with our HDF5 or ZAR backends, depending on your needs. This standard organization is great for data types that are commonly used across the field. But as you know, neurophysiology is constantly innovating new methods. So how do we ensure those new and specialized data types that might not be supported by the core already can be stored and shared as well? In those cases, you can create an NWB extension to describe your new data type and the metadata for it. This is particularly useful for when you want to store data and metadata for a new method, for acquiring or processing data, or you want to store metadata that's specific to your lab or your experiment. If you use an extension the extension itself is packaged with the data so that other people and other software tools, they can understand what your new data type is and how it's structured. And you can find over 30 extensions developed by the community in the NDX catalog at that link over there. And you can develop and share your own extensions in the catalog as well. So here's a few examples of the 30 extensions in the catalog. Most of them add support for new acquisition types and experimental metadata. And I want to emphasize that NWB is not a static data standard. It continuously evolves to meet the needs of the community through a community-driven process. So major changes, such as integrating the extensions that I mentioned earlier into the core standard, will go through a formal NWB enhancement proposal process, or NWB EP, where proposals are submitted, they're reviewed by the Technical Advisory Board, and then they're evaluated by a broader group of community stakeholders outside of the initial proposers. And this keeps NWB current with the latest methods and data types in neuroscience. Right now, we have 4 proposals currently active, with a handful more that are being planned. Proposal 1 has just been accepted and integrated into the core software. This adds core support for core and consolidated support for event types in NWB. Proposal 4 is currently under community review, and we'll do a bit of add extra support for storing metadata about different optical and microscope devices. As part of proposal number 1, we're also adding support for annotating events with a standard called hierarchical event descriptors, or HEAD tags, which is a controlled vocabulary for describing experimental events. And this allows scientists to describe their stimuli and behavior data more formally, and enables tools downstream to better interpret and process these data. So I just described the rules of the standard, but how are these data structured on disk? Internally, NWB uses HDF5 as its primary file storage backend. HDF5, it's a popular scientific data format used in many scientific fields for large-scale array data. It supports advanced I/O features such as lazy loading and ways to optimize your data storage and performance using chunking and compression. Oliver will show how the lazy loading will work in his talk. HDF5 is also well supported across computing platforms and programming languages, and it has long-term support, so your data will always be readable. I think HDF5 has existed for 20 or 30 years now. NWB also supports ZAR as a new alternative storage backend. ZAR is relatively new, and it's more optimized for cloud computing, and we're working with the community to explore the main use cases for that and to support that as best we can. We're also working with labs to explore and support integration for other data management and storage systems such as data joins. Okay, so what I've explained so far is NWB as a data standard. These are the rules of the system. These are how the data are written to disk. But NWB is more than just a standard. It provides a suite of software tools that work together to make it easy for scientists and developers like you all to work with NWB data. So we provide core tools for converting data to NWB, which I'll describe next. We also provide APIs in Python and MATLAB that allow you to easily create, read, write, and use NWB data. I'm sure you all will be using either PyNWB or MatNWB through this week. And we're also working on a C++ API called HackNWB to enable data acquisition systems to write directly to NWB, saving you the conversion step. We provide tools that aid in the development and sharing of extensions, and we provide some other important lower-level tools that support the standard and software. So we've done a lot of work here over the past 10-ish years, and so if you're interested in learning more about the technical stack, please chat with me. So now that you have a better idea of what NWB is, you might be thinking, how do I get my neurophysiology data into NWB? And we'll later get into how do I read and reuse data. I'll have a more dedicated talk on this in a few days, but just to give you a high-level overview. So We know experiments are quite diverse, they generate a lot of data, and they involve combining data from several different modalities. You might have a neuropixels recording from a spike GLX system, plus the sorted units from that recording, plus the raw video data, plus pose estimates from that video data, and stimuli, et cetera, and so on. So how do you get all of this into NWB in the first place? So we provide 3 sets of tools at different levels of accessibility and flexibility. NWB Guide is our no-code conversion app that you can use. It's kind of, we think of it kind of like TurboTax for those in the US. It's basically extended form entry where you point to your data on your system and you don't have to write any code. You just enter it and it will do the conversion for you. NeuroConv is a kind of an intermediate, involves some code and works for most acquisition formats out there. And we also provide the Python and MATLAB APIs where you can use custom code to handle reading and writing of your data. So just to step a little bit into more detail into these 3 options, NWB Guide is a desktop app, provides you this graphical user interface for you to fill in the details. You basically select your acquisition system. If it's one of the 40 that are supported, it will read the metadata from those files and the data from those files, and it will validate the metadata and prompt you to fill in any missing metadata. And then it can batch convert all of your data from all of your sessions to NWB and shows you interactive visualizations of your converted data so that you can verify that the conversion was correct. Finally, you can use Guide to upload your NWB files to the DANDI Archive, and you can do all of this without having to write any code. Guide works for converting data from certain supported data formats to NWB. If it doesn't support your particular format or you want to convert data programmatically, we recommend that you try NeuroConv. NeuroConv is a Python package that converts data from even more formats to NWB and provides more options for customizing the data structure and the I/O. Conversions generally require a small amount of Python code. So for example here, converting a single spike GLX recording to NWB using NeuroConv takes about 10 lines of Python code, including the imports. Then the last line there is just run the conversion, pointing to the destination NWB file path. We hope that this is useful for those of you who are converting data in your labs. So here's a full list of all of the formats accepted by NeuroConv. If your favorite format isn't on this list, let us know, or create an issue on the NeuroConv GitHub repository, and we'll work with you to support it. We'll ask you for some sample data, and we'll figure out how to read it and get it into NWB. And finally, we provide APIs in Python and MATLAB that allow you to read and write NWB data. These are useful if you're working with data that's not supported by Guide or NeuroConv, such as a custom MATLAB file, or you have trial data in a custom spreadsheet, or you're using extensions that only you know about, or you just want really fine-grained control over how the data is laid out. We have a ton of documentation and tutorials for how to use these tools at these links over on the right. And the tutorials, they range from the basics of reading and writing to advanced topics such as customizing your compression and streaming from the web. Okay. We're also excited to show off our new conversion chatbot that's integrated with the PyNWB and NeuroConv documentation. Let's see if this works. You just go to the PyNWB docs, and over here on the right, you can click Open Assistant, and you can ask it a question like, how do I convert my Spike GLX and Phi data to NWB? And it has indexed all of the NWB documentation and NeuroConv documentation, and will kind of guide you through step-by-step the process. And you can chat with it with your specific use cases. So there's the code, and you can update it as needed. And citations so you can verify that it's correct. This is pretty new, so we'd love your feedback on it if you use it. Yeah. Okay, and just a brief plug to say that we're developing HACK NWB, a C++ library for recording data directly to NWB from acquisition systems. And we're working with systems like OpenEFIS and BlackRock to integrate this so you can get NWB files straight from the box to NWB without needing to convert afterwards. Okay, so now I'll talk about data sharing and reuse through the DANDI Archive. Again, this will be a brief overview. Ben will talk more about this in his talk. So the DANDI Archive, dandiarchive.org, if you're not already familiar, is a platform for sharing and analyzing neurophysiology data. Most of the data on here are neurophysiology data, but there's also some static microscopy data, ex vivo MRI, some light sheet microscopy there as well. DANDI accepts neurophysiology and associated behavioral data only in the NWB formats. And so that when you contribute data, the DANDI will automatically validate your NWB files, check that it follows best practices, and then automatically extract from the NWB files metadata that it needs to populate its DANDI pages. And also enables DANDI to allow complex searches of data on the archive. You can upload hundreds of terabytes of data to DANDI for free. And this is because DANDI works with the Amazon AWS Open Data Sponsorship Program to provide free storage of open scientific data. This means that DANDI can handle all of the raw data for most experiments, and we encourage you to upload all of your data because it may be useful for other researchers studying questions that use raw data, or may be useful for people developing new methods like for processing data, like spike sorters. We just ask that you talk to us or the DANDI team if you want to upload more than 10 terabytes at once, because we want to make sure that you're storing data correctly, using good compression, and following best practices. You can also upload your data in embargo mode, which means that your data is accessible only to you or your collaborators until you decide to publish it, or you publish a paper with the data. So there is a eventual, this data needs to be published, but for some amount of time, you can keep that, you can publish that data on DANDI and have it just accessible by your group. Once your data is in NWB, you can publish it on DANDI, and we recommend that you use Guide to publish your NWB data. You don't need to have converted your data to NWB using Guide. There's a standalone upload option available. You can also use the DANDI command line interface provided by the DANDI team to do this programmatically. As Ben mentioned, there are now over 550 NWB datasets publicly available on DANDI, and this totals 500 terabytes of NWB data. So that's a lot of data for you all to explore and work with this week. Most datasets on DANDI are called DANDI sets and are from the most common animal models for neurophysiology. So mice, rats, monkeys, humans, etc. But DANDI also has some datasets from cattle and even from ants. So modalities can vary across datasets, with over 200 datasets containing extracellular EFIS data, over 100 with behavior data, and same for over 100 for microscopy, such as 2P and 1P imaging, and over 50 datasets with intracellular EFIS data. I think these numbers may be a little out of date, but so there's even more nowadays. There's so many interesting datasets. I just want to highlight a couple large-scale survey data sets that you might be interested in exploring and analyzing this week. There's the microns data set. This is DANDI, well, you can just search microns. It's functional data from thousands of neurons in the mouse visual cortex that are co-registered with EM, electron microscope data, and a map of their synaptic connections. So very rich data where you can really explore the connections between function and structure. Function and connectivity. There's also the IBL, brain-wide map. This is recordings from over 200 brain regions, and Pranav will talk about the IBL datasets later today. The Allen Institute, a huge proponent of open data, has published over 35 data sets on DANDI, and this includes the BICCN and BICAN consortia data sets, the visual coding and visual behavior data sets from the Allen Brain Observatory, the SHIELD project, and the OpenScope program that Carter and Jerome will talk about later today, and hundreds more. And as a preview, I just want to show you the first 10 data sets from the OpenScope program. The data was designed to be reused from the start, and so it's processed through a common data processing pipeline and is of consistent high quality. And these are mostly vision and behavior data tasks in mice, and include both extracellular ePhys data as well as 2-photon imaging data. And the OpenScope team also provides this awesome OpenScope data book alongside these data releases. And this data book is an online tutorial and executable textbook on how to visualize data from NWB files, perform first-order analyses and more complex analyses using the OpenScope data as an example. The data book also includes a comprehensive page on each of the experiments that they've published that demonstrate how to analyze and explore this data, and even have some exercises so that you can use it for educational material or learn from them. Okay, and we had a question about this earlier. In addition to being a data archive, until the end of the month, DANDI also provides a free Jupyter Hub instance that provides fast access to stream and download DANDI data because it's hosted in the same region, the same AWS region as the data itself. So you can analyze data sets in Python on DANDI Hub. It's kind of like Google Colab as well. And I believe right now you have access to different types of servers, including a GPU. You can also share and find analysis notebooks for different data sets on DANDI Hub. And because each DANDI set is a different experiment, it contains different kinds of data, if you're interested in reusing that data set, it's often helpful to see how someone else opened the data and made sense of it. And I think Ben will walk people through this page later today. So many of these notebooks were created by the dataset owners, but some were created by community members. And so as you are going through this week and exploring how to open and analyze existing data, we hope that you will encourage you to publish your notebooks to this repository by the end of the week to help the next cohort of data reusers. Okay, the easiest way to find data on DANDI is just using the search bar. You can type neuropixel or hippocampus or Parkinson's and you'll get a list of relevant results. You can also search programmatically using the API for more complex queries such as find all NWB data with at least 10 sorted units from area CA1 in male mice less than 6 months old or something like that. That's doable with the programmatic API. Okay. And as Ben mentioned, with over 550 NWB DANDI sets, we're starting to see these data being reused in publications. We found over 150 papers that reuse NWB data for secondary analysis, tool or method development, and much more. So I think Ben went through some of these examples already of data reuse, including scientific discovering new science in this paper by Farrell et al. Discovering a new relationship between hippocampal dentate spikes and elevated firing rates across the brain. The Mathis group published on Zebra, learning latent embeddings from both neural and behavioral data, and this figures directly from NWB data on DANDI. Ava Dyer's group has trained foundation models on multiple large-scale neural data sets, some of which are hosted on DANDI, and Kilosort showcases their new method on multiple DANDI sets. DANDI is being downloaded from across the globe, so we're really starting to see the broad impact of data standardization and data sharing. Okay, So finally, I'll showcase some of the tools that have integrated with NWB to help you with analysis, visualization, and data management. Okay, so I showed this before. This is kind of the core NWB schema at the bottom, the core API tools on top. We maintain all of this. This creates, on top of this, well, what we've built creates a foundation for people in the community to develop a variety of awesome tools to explore the data and analyze the data. So we have a bunch of tools to explore data, such as NeuroSift, which Jeremy will talk about. There's also NWB Explorer for intracellular EFIS data, and HDF5 tools can work well with NWB data as well. NeuroSift is developed by Jeremy Maglin at the Flatiron Institute. It provides interactive visualizations of data based on neurodata type, and it streams the data selectively from the archive. So even if your file is 100 gigabytes, NeuroSift streams only the parts that you're trying to plot and not the whole file. It runs in the browser. You don't need to install anything. And it's integrated into DANDI and also NWB Guide, so you can explore unpublished NWB data on your local computer as well. So I'm going to do one demo, one demo of trying NeuroSift on this data set's 582 conjunctive representation of position, direction, and velocity in entorhinal cortex. This data set comes from Sargolini et al. 2006, one of the key papers from the Moser lab that supported the discovery of grid cells in the medial entorhinal cortex and contributed to the Mosers winning the Nobel Prize. So we're very fortunate to have this data set up on DANDI for you all to play with and explore. So you can, I'll just go to just show you how to do search as well. What data set was this? We'll look for entorhinal cortex. 10 results, and we are looking for this one here. DANDI set 582. You can go to, Ben will show you more of this. You can go to files and go through each subject to access individual NWB files. And so we'll just look at the first one here and open this with NeuroSift. You can also download this to your computer to explore. But we're going to showcase NeuroSift. If you do this, this will load in the NeuroSift viewer, which shows you the hierarchical organization of the NWB file. And each of these are clickable to explore what the data looks like. So you can click a little button here which will show you the electrical series recorded from, let's see, these are EEG signals from one electrode in, I forget which, medial entorhinal cortex. And you can, how is this going? You can scroll through the data. You can zoom out. Is the zooming working? Yes. And all of this data is being streamed directly from the DANDI Archive into your browser. You can also go to and select, let's say, the LFP and the XY position of the animal and plot those together. Let's see, I think that's right. Yep. And let's just zoom out a bit or zoom in a bit. And then you can scroll through time and basically look at neuroactivity in the medial entorhinal cortex as the animal is going through different parts of this open field. And you can use this to figure out whether this data is appropriate for your study, and then use it to, and you can download the data and really try to replicate the original Moser experiment. And I think Guillaume will showcase this in his pineapple talk in a few days. Okay. Great. So We have a handful of tools for data management and publication and analysis. This includes Pineapple, where the data are input as NWB files, and then you have access to a number of methods that do aggregation, do time filtering, or time restriction of time series data. You can do thresholding and many other things. And you can do much more complex things that build on these basic methods, such as peri-event time histograms. You can compute tuning curves, for example, to look at the grid cell map from the Moser lab. You can do feature decoding and cross-correlations of spiking data as well. And Guillaume will go deep into this in a few days. You can also look into using the Zebra method. This is developed by the Mathis lab, and we have a notebook from NeuroDataReHack 3 years ago demonstrating how to use Zebra on some data that was some NWB data on DANDI to produce latent embeddings of a particular data set. You can go to the Zebra websites to learn more and explore some of their other tutorials. We also have tools that work well with extracellular ePhys data in NWB. This includes Spike Interface. This is a Python package for creating and evaluating spike sorting pipelines. Using Spike Interface, you can basically load your data from a variety of recording formats, such as including NWB, and you have access to, I believe, 8 or 10 different spike sorters. Different versions of Kilosorts and Mountain Sort, Spiking Circus, Try to Clue, Iron Clus, etc. And you can run all of those spike sorting algorithms on your single data set. You can use them. You can use this to compare the spike sorting results and compute metrics and evaluate them against each other. Alessio will be talking much more about this in a few days as well. Cell Explorer is another tool developed by Peter Peterson. It provides a graphical user interface for exploring and classifying single cells based on their waveforms and spiking statistics, and very nice tool for exploring extracellular ePhys data. Okay. There's also a handful of tools for intracellular ePhys data, such as MEES, PatchView, BMTK, just showing you what PatchView looks like. It's a graphical user interface for data analysis and visualization on multi-channel whole cell multi-patch data, and allows you to do things such as firing pattern analysis, event analysis, morphological analysis, etc. And you can go to this website for more details. We also have a bunch of tools for optical physiology. You might be familiar with Cayman and Sweet2P for calcium imaging processing. Cicada, Robin will talk about that in a few days. That allows you to build more complex analysis pipelines. Optimist also allows you to do that. And Cheetah and Extract are also other ways for processing 2P data. And lastly, there's a number of tools that work with behavior data in NWB. So you might have heard about Deep Lab Cut and Sleep. These are tools for estimating the pose of animals through movies and machine learning. And both of these tools can write data to NWB. Write the pose data, and in the Sleep's case, also writes training data to NWB. Okay, so you can explore all of the core tools and all of the analysis and visualization tools from the community on our website, nwb.org/tools/core or /community. We encourage you to go there to learn more, explore, and go to links to all these different tools. Okay. And I want us to mention that most of these tools use NWB as both an input and output format. So you can build data analysis pipelines where you can easily swap one tool out for another, or build complex pipelines that chain these tools together to speed up your data exploration, processing, analysis, and management. All right. So I know that I just went through a lot of information. So I want to make sure you know how to get help when you need it, especially after the workshop. You can ask us questions directly on our Slack workspace or our NWB help desk. You can just go to bit.ly/NWB help desk. This is a GitHub discussion forum, and we actually prefer that you send questions our way through the help desk because it's public and therefore indexed by Google, so that other people who might have the same question can find answers there. And you might find an answer to your question there as well. We can also set up one-on-one Zoom consultations, and we encourage you to join our newsletter. So go to this website, bit.ly/nwb-email. I think there's also a link on the main nwb.org website. And we send maybe 3 or 4 emails per year announcing upcoming events like this, any software updates, and other news relevant to the community. If you have trouble with DANDI, you can ask questions on the DANDI help desk or email them directly. We also have, you can also explore our extensive documentation on the nwb.org website and previous recordings of talks on our YouTube pages. Okay, so going back to the original starting point, looking at the state of data sharing in neurophysiology. So, hope to have convinced you that all of these different ways that NWB and DANDI help enable data sharing and reuse. So sharing NWB data on DANDI really extends the life and impact of valuable data sets. And this ultimately will accelerate, we hope, scientific discovery across the field. NWB data can be understood and reused by anyone familiar with the standard, and the DANDI Archive provides a platform for publishing and finding NWB data. So we hope that this will help make reuse of NWB data much easier and can therefore drive collaborations and the development of new science. And finally, data in NWB can be read by a large number of community tools. Because the data follow a common standard with metadata standards, these tools can rely on the data being formatted a certain way, can rely on metadata being present that they need to do their analysis, and it makes access to all of these tools much easier for all of you. NWB compatibility also enables the chaining of all of these tools together in whatever way suits your research best to accelerate your science. Okay, so stepping back again, our goal with NWB and DANDI is to help labs overcome the energy barrier of data sharing and get more and more scientists on the right side of this graph, sharing and reusing data. And during this workshop, you'll be working almost exclusively on the right side, where you're taking advantage of data that's already been standardized and shared in order to discover new scientific insights. And then this is important not just for advancing your own scientific interests, but also to help demonstrate the value of sharing all of this data in the first place. With that, I want to thank our sponsors, our funders, the NIH Brain Initiative, the Kavli Foundation, the Simons Foundation, Michael J. Fox Foundation, for all of their supports over the past 10 years of both of these projects. And I want to also mention that NWB is truly an endeavor in interdisciplinary team science. Neither NWB nor DANDI would be possible without the contributions and support of everyone in the community. All of our work is open source, and so we invite you to be part of it, contribute to the code and documentation, join a working group, lead a training event in your lab, or invite us out to lead a training event at your institution. Spread the word to your peers about what you learn throughout this workshop. Even if you don't get directly involved, your feedback guides our development priorities. So please let us know your thoughts on the current state of the ecosystem and what you would like to see. How would work on our end benefit your research? Our teams ultimately exist to serve the neuroscience community, to enable open science, to drive collaboration and data reuse, and ultimately advance scientific discovery. And we're super excited that you all are here to learn and be part of it. So with that, I want to thank you all and happy to take any questions. can we get the mic? Could someone throw a box? Great.
So you shared like this NDX, which is like you can define your own format. Is there any restriction in that, or you can come up with any kind of data organization for that NDX format?
It has to follow the NWB specification language of there's certain of there's groups and data sets and attributes and links, but how that is all structured for your new data type is up to you. It has to also fall into the larger hierarchy, kind of broader organization of an NWB file generally. But you can define a new type for fiber photometry. You can define a new type for 3P imaging or whatnot that we might not have covered in the core. And that can be like many levels deep with like hundreds of parameters that describe your specific acquisition system or your specific experiments.
I will say usually you start off with picking some base type that exists in the NWB standard, right? If you do a time series recording, you're not going to define a completely new time series type. You say, I'm building off a time series, adding new information to it, or I'm refining it. Or, if I need a data table, I'm going to start off with the table data structure that I defined, and I'm going to refine it, right? So typically, that's how you fit into the standards, by sort of picking off the pieces that are relevant to you and sort of refining them and building on them. But you can obviously build completely new structures as well, but typically the idea is you want to be as close to the principles that exist and then sort of refine and build on those extensions.
Yeah.
When you upload your data as embargo mode, is there a time limit to keep it like that? Or you can have it embargo indefinitely?
I'll let Ben answer.
Yeah, okay. Good question. So the DANDI Archive was created for NIH data. Anybody can submit data, but if you do, you have to follow the rules of the NIH. And they have a new data sharing policy that says when you publish a paper, you have to share the corresponding data. So or if a grant expires that funded the research of that data, then at that point you should, if you haven't already, at that point you should share the data. So we have data contributors enter the grant number and the expiration date of that grant, and then they're expected to open it. Now, we don't automatically do that at this point, but we might change that to where we're really enforcing that. But if there are projects that don't have like a clear funding with a grant number and an expiration date, then we just default to 2 years. Yeah. So so basically everything, gets out there within a few years. Yeah.
Any other questions on NWB and DANDI?
Go for it.
So with such an extensive suite of tools that build off NWB, does that make it very challenging for your team to make the updates you were describing on one of the slides? Like you had, I think, a list of 4 things. Like, do you have to go and test that these updates are compatible with like 30 different extensions and tools? It
is a challenge, but we try not to make breaking changes in the software. And when we do, we try to give ample warning to the developers of downstream tools to make sure we're not breaking anything in their ecosystem. In the Python world, you can also pin your software dependencies to specific versions of PyNWB so that it limits any impacts of major breaking changes. But that's something that we think about a lot, is try not to break everything all at once, because that's not great for the community.
It's also when NWB as a data standard, right? our goal is to ensure that, data remains accessible in the long term.
Yes.
So as we're making changes, we're also testing against older versions of the data to make sure that that data remains readable, that we're not, just because we're adding a new feature in 2.11, that the old data doesn't become obsolete, right?
Right.
That is really a major goal of us, that data that you publish remains accessible.
Right. I was talking just about the software, but for the data itself, all NWB 2.x data are readable by our Python and MATLAB APIs, and that goes back the history of the project. Okay. Thank you all. Before